I thought that this would be a special subject that we should talk about for a lecture. Okay. In terms of the history of antibiotics, um, antibiotics were developed and first used in World War II, and this is a poster that was out at the time that says, "Thanks to penicillin, he will come home." And it's very true that being able to cure infections from war injuries was one of the major things that helped, or that having antibiotics was a major thing that helped keep the fatalities in the Second World War. Okay. And when antibiotics were first discovered, they were known as a magic bullet. And what they were saying with the magic bullet concept was that it would kill the pathogen but not harm the host at all. And so what you have to do is you have to take advantage of the differences between bacteria and the host so that you don't get killing of the host at all. Okay, so you only want to be able to kill the bacteria in this case. That's a sort of easy way of saying selective toxicity. And selective toxicity is just simply being able to kill the pathogen and not the host. And when we're thinking about antibiotics, we can use a selective toxicity to figure out both the therapeutic dose and the toxic dose. So in the therapeutic dose, this is the amount of drug that we need to kill the pathogens but doesn't harm the host. And you can also have a toxic dose, which would be the level that causes damage to the host. And here are just a few possible toxic side effects to the host for different antibiotics. You can have types of anemia, kidney damage, tooth blackening, sleep and mood disorders, and that last one, the upsetting the balance of the normal flora. The normal flora are the bacteria that live in and on you, and we are in a pretty big relationship there where those bacteria are helping the host survive. But if you take antibiotics, you could be upsetting that balance, and so something could, while you may be taking the antibiotic to get rid of one pathogen, you may be taking care of or killing a lot of the normal bacteria that you don't want to happen. A little bit more history. Paul Ehrlich is the first person who discovered anything in an antibiotic, and he reasoned out that there would be some chemical agents that could be selectively toxic so that it only killed the pathogen and didn't kill the host anymore. 1904, he used the compound trypan-red against trypanosomes. Trypanosomes are a parasite involved in African sleeping sickness, and that was pretty much the first case of trying to use this selective toxicity. Haida was a co-worker of Ehrlich's, and he developed arsenic derivatives that could specifically kill syphilis. Dogmark used a compound called Ponicil Red, which was safe to humans, but definitely killed Streptococcus and Staphylococcus. He received the Nobel Prize in 1939 for that discovery of Ponicil Red. Now, we sort of always think about penicillin being discovered by Alexander Fleming, and there is some evidence to suggest that it was first discovered, penicillin was first discovered by a med student in 1869, and because he was just a 21-year-old med student, people ignored him. Okay, it wasn't, it didn't seem like he could have made such a great discovery, so that discovery just kind of went without people really noticing it. So in 1928, Alexander Fleming looked at a plate that he was doing some experiments with Staphylococcus on it, and he noticed that the plate got contaminated with a fungi, and around where the fungi were, the bacteria, the Streptococcus, weren't growing as well as those near the fungus. And here's a picture of Fleming's original plate, where here you have the mold Penicillium colony, and then here are the normal colonies, and you can see that they're a lot smaller the closer you get to this edge of the plate, and so of the mold. And so he reasoned that there must be something from that penicillium colony that is causing the bacteria to die. Here's another more common or modern picture of a very similar type of experiment, where we have normal colonies and then this very large mold colony. And in this case, you can actually see some of the compound is it's a little bit colored, and you can see that ring around it. And wherever that compound is coming out, the bacteria don't grow as well, whereas out farther from that, they seem to grow just fine. Okay. Now, Fleming started trying to isolate the compound, but he wasn't really very effective at it. Okay, he really couldn't figure out exactly how to get the active compound out of the penicillium bacteria. So two people, Florey and Chain, who were at Oxford, started to work and used this compound, or used a new kind of technique to be able to isolate just the active compound. And it took Florey and Chain quite a while to get a decent amount of it, but they did receive Fleming, Florey, and Chain did receive the 1945 Nobel Prize for the discovery and purification of penicillin. Someone else who was early in the discovery of antibiotics was Sal Waxman. And Sal Waxman screened over 10,000 strains of soil bacteria, and of those he found one, Streptomyces gregis, that produced an antibiotic. In this case, he made the active compound named streptomycin, and he also won a Nobel Prize in 1952 for this discovery. How Waxman did this work is he would take one known type of bacteria, so that's this line here going up and down, and then inoculate different kinds of bacteria from right to left on this, and you can see these are very specific bacteria that are each of these lines, And what you can see that this antibiotic kills a lot of the Streptococcus epidermidis. Okay, you have pretty good growth up here, and you have no growth down here, and these other ones don't really inhibit whatever this bacteria that he's screening for. It didn't produce anything that was killing these other bacteria. And so when I said that Waxman used about 10,000 of these, he literally did this up and down streak on 10,000 different plates, and then did the cross streaking with different kinds of bacteria to try to find something that had the inhibition like the Streptococcus epidermidis. Here's a list of some of the more common antibiotics and the bacterial species that they come from. The vast majority of these streptomyces, which look like molds, and to a first impression look like molds. And then we also have some bacillus species. I just want to point out one of the things. Under these two bacillus species, we have bactracin and polymyxin B, and up here we have neomycin, and neomycin, bactracin, and polymyxin B are the common things of the components of neomycin cream. And what part of the reason I point that out is it's a whole lot better for you to buy the triple antibiotic ointment at the drugstore as opposed to neomycin, because the neomycin is pretty expensive. You get a few ounces in a tube for X amount of dollars, but for the same amount of money, if not cheaper, you can actually get a big tube of triple antibiotic ointment, and it'll be exactly the same active ingredients, the neomycin, the bactracin, and the polymyxin B. It's just that you get a whole lot more for your money. Okay, we can divide our antibiotics into two broad categories. The first is the broad spectrum, which will kill both gram-positive and gram-negative bacteria. And then there are narrow spectrums, which only kill a single group of bacteria. So when you look at this chart here on the bottom, okay, something that can kill, say, a whole lot, both gram-negatives, gram-positives, and some of the obligate parasites, tetracycline would be a really broad-acting, broad-spectrum compound. Okay, something like vancomycin is a narrow spectrum because it only kills gram-positive bacteria. Okay, and that's in part how the antibiotic kills the cells. So there are advantages and disadvantages. In a broad spectrum, you have a better chance of getting what's ever infecting you to die, but there's also, you run the risk of killing off more of the good bacteria that you want. And with a narrow spectrum, one of the big problems there is you might try the wrong antibiotic, and so that's going to keep you from killing the pathogen that you're really concerned about. Okay, here are the targets of action of different antibiotics. And the big take-home from the whole slide is that you're always going to have the antibiotic working on some essential function of the cell. Okay, so whether you need to make more RNA, whether you need to be able to make DNA, be able to make proteins with protein synthesis, any of those essential functions are those things that you're going to target with your antibiotic. And it's not important to, say, memorize all the different drugs, but what's important is to know that these parts of the cell that are essential to the cell, that those are likely candidates for the antibiotics. Okay. Now, one specific example is with penicillin, and of course we learned about penicillin as the very first commercially available antibiotic. And what penicillin does, it inhibits the cross-linking of the transpeptidase bond. And so as you're making a cell wall, you have to have a number of bonds of the cell wall, and this is a very specific bond that gets broken. Now, the structure of penicillin, this structure is kind of a generic penicillin, but the most important thing is this beta-lactam ring. Okay, so it's a square ring with nitrogen and 3 carbons, and this is the kind of unique structure that makes this effective. You can have a lot of groups on the outside, things on the other side of the molecule that will all help get the drug to the wound or whatever, but it's that beta-lactam ring that's going to be doing all the killing. Okay, and here's just a structure of a cell wall and where the inhibition works. So circling the cell, you have a series of carbohydrates. Okay, these are sugars that are linked and circle the entire cell. Then from the sugars, you have small peptides, and the terminal step in connecting one amino acid to another amino acid, this is the place where the beta-lactams bind. And when that can't be made, so that we now have no peptide there, or no peptide bond there, the entire structure becomes really weak. And in general, it's not going to be just one inhibition like this, but when at the therapeutic dose, there are going to be a lot of these bonds being inhibited, which will keep the cells from staying together. There are a number of different derivatives of penicillins, and you might know this, well, it's all in that acyl group. Okay, when I showed you that structure. And then it will also a little bit depend on how you take the antibiotic. So if the y-axis is the concentration in the blood versus the number of hours after taking it, you can see something like penicillin G, which is injected right into a muscle. That's going to have a very high rise and be at very high levels very quickly, but it'll go away fairly quickly also. Okay. The same antibiotic, but just taken orally, will only go up to a very moderate level and will go away a little bit slower, but still the levels aren't quite as high. Okay, and so this is one of the reasons why you might always want to get a penicillin shot instead of just taking the penicillin orally. There are definitely antibiotics or penicillin derivatives that can last for a very, very long time, and you have to worry about how much of a dose there is, because you again have to get to that therapeutic dose, or else the antibiotic isn't effective. Here are it's a tree of the different penicillins and or different penicillin derivatives, and when they were discovered or invented. So the original, right, that you would get right from the mold penicillium is penicillin G, and it was one of the very first compounds that was ever introduced as an antibiotic. And then you have a whole lot of other derivatives where we're changing that acyl group, that group on the outside of it, which makes them more effective, easier to get into the bloodstream, things like that. And so, there are a large number of penicillin derivatives, and they all have some special property to them. So doctors are constantly trying to find which antibiotic is best for you, and which of the penicillins even will serve your needs. Okay. These two images show you some of the effects of penicillin. And over here you can see a nice cell wall here. Okay. And then you have this little bulge. Okay. And a nice cell wall here. And what's happening is that because the cell wall has been weakened, the insides of the cell, the cytoplasm, is able to just bulge out part of the cell. Here's another example, and in this case, we actually see a tearing away of the cell wall. So here we can see cell wall, some cell wall here, some cell wall here, and then all this are the guts of the cell just sort of vomiting onto the microscope slide. Okay. A drug that we can't take internally, but we can use on the skin very well, is called polymyxin B. And polymyxin B is actually going to be able to insert itself into the cell membrane, and it'll change the charge. Okay, so you might see that the if this is the outside leaf of the lipid bilayer, and it is normally quite negative, polymyxin B gets inserted in there, and we now add a bunch of positive charges. And so the polymyxin B is altering the permeability of the membrane by introducing the opposite charge. Okay, this is one of the most important things I hope you learn out of this class, and something I hope you take from this course. And that is to take your medicines for as long as prescribed. So if you look at day 0, you'll have a population of biology that the majority of them are an average intermediate susceptibility to the antibiotic. You'll have a few that are highly sensitive, and you'll have a few that are a bit more resistant. Now, this is not antibiotic resistant already. Okay, it's just that for killing that specific organism, they don't like to be killed as much. Okay, and again, this is a nice even distribution across at day 0. Now, by day 3 on the antibiotic, you have killed off all the susceptible ones. You've killed off about half of the intermediate ones, but these ones that are higher resistant, you haven't really touched that population yet. And the 3 days is a bit significant because let's say you have a cough or something like that, and so you put on antibiotics. After 3 days, because you've killed so many cells, you start feeling better. Okay. If we were to continue to take it till day 6, we'll get rid of almost all the intermediates and the low bacteria, and then you still have a population of some of the more resistant. But if you take it for the full 10-day course, you'll get rid of essentially everything. Okay, and this is really important if you're diagnosed to take an antibiotic for a certain number of days, and certainly the time that you how many times a day you need to take it. You need to do that for the full course, and try not to skip any doses, and always take it till the very end of the course as prescribed. Now, here, let's say that after day 6, you decide to stop taking your antibiotic. Okay, so you've gotten rid of most of the bacteria, but you still have those most resistant ones still alive. And if you stop your antibiotic before the entire course, then this smaller population of the resistant ones will grow, and they will either be just a greater number of them, or some of them may mutate so that they actually become completely antibiotic resistant. And that's one of the places where you worry about something like this, that you can't have, you don't want to develop antibiotic resistance because you didn't take your antibiotics correctly. So in general, I want you to always take your antibiotics for as long as prescribed and at the times that you're supposed to have them. Okay, so if it's just once a day, then you'll just take it one time. But if it's 3 or 4 times a day, make sure you space those out evenly and take them equally. And don't miss a dose. Antibiotic resistance is a big problem and something that we're having more and more problems with. And when I'm talking about resistance now, these are mutations that occur in the bacteria to allow the bacteria to metabolize the drug in a couple of different ways. And so this antibiotic resistance is a true mutation, and one way of thinking about it is that you have gotten the bacteria to evolve into something that's that is resistant to it. Okay, the important thing about these mutations is that the antibiotic doesn't make the mutation. Okay, the antibiotic making the mutation would imply that we would damage the DNA in some way so that the antibiotic was actually causing the mutation, but rather there are just random mutations that occur whenever a bacteria is replicating, and when you have an antibiotic around, you do is you select for those, so you're increasing the number of those resistant bacteria because you've selected for them. You've given them a growth advantage in that environment. And the mutations that the bacteria get can do a couple of different things. First, they can break down or modify the drug. The penicillinase is an example of that. That beta-lactam ring that I showed you is key, and if you break that bond, then the antibiotic is no longer effective. We can have pumps that can export things from the cell. And finally, we could tighten up the cell wall to keep the drug from actually even entering the cell. Now, antibiotics resistance costs us a whole lot in terms of healthcare, and the estimates are pretty wide, but it's on the low end. People think that it might cost the U.S. $100 million a year, and some studies will say that it can almost cost $30 billion a year. So while a big range, I think the easy take home is that there's no doubt that antibiotic resistance costs this country a lot. Well, impossible to kill with that antibiotic, and depending on which antibiotic, which kind of bacteria, it might be impossible to kill anything or to kill them with any antibiotic. So when I was saying the mechanisms of all the antibiotic drugs, the drug has to be able to get into the cell, bind with its target, and then that complex will go on to kill the cell. Now, when you have antibiotic resistant cells, okay, one of the things that's going to happen is that you're going to one mechanism is to take the drug and modify it and inactivate it. Okay, so now it can't get to the target because it's not the same shape anymore. We can also alter the target so that there could be something that's in the cell that you want to use to kill, but once you change the shape, this antibiotic won't be able to bind to this anymore. Now we can have things that will pump the antibiotic out of the cell. Okay, this is a passive coming in, and then the antibiotic can be actively pumped out of the cell. And we can also have a decrease in the uptake. Okay, so this antibiotic wants to go into the cell, but something has changed. Some protein on the surface has made it so that it can no longer accept that drug into the cell. Now remember I said that antibiotics don't cause the mutation, but rather they're selecting against things that have a mutation. And it's fairly dramatic just by looking at the amount of antibiotics used and the number of those strains that are resistant. Okay, so if you look on the left side, so these are the tons of antibiotics used. And remember, you're mostly taking antibiotics in the milligram quantities. So these are worldwide tons of usage. And some of these don't have incredibly high numbers yet, right? Maybe nalidixic acid might be about 10% of the population. But some things like tetracycline are real close to 80 to 90% of those bacteria are actually resistant to tetracycline. Okay, it's almost making it to the point where tetracycline can't be used clinically because there's so many things that are already resistant to it. Now, one of the places and one of the ways we get such high tonnage of chloramphenicol is that it's very often given to dairy cattle to help them maintain and put on weight while they're growing. And so a number of different animals are given antibiotics so that they put on more weight so you can sell them for more. And there is a little bit of evidence that some of the antibiotics are going to remain in the animal. Some animals and some antibiotics just won't, but there's definitely a push to have meats that are all antibiotic free. And the problem with that is that since the farmers will lose some of their animals to disease, they have to charge more for those things that are grown without any antibiotics. One of the somewhat nice things about antibiotics is they're very cheap. Okay, so something like the tetracycline that might be put in to cattle's feed so that they're eating it, it might be a few cents a day to be able to feed 100 head of cattle. Okay, and so it's definitely worth it to the farmer because that cow is going to be able to be sold for more weight at the end, and they've only had to invest a few cents to be able to get that weight up. In panel B, this is a little bit scary too, and so here's the percent of strains of Neisseria gonorrhoeae that are resistant over years. And one of the reasons that this graph becomes extra scary is Neisseria gonorrhoeae is the causative agent of gonorrhea. Okay, and you can see that about getting up to 10% of the cases that are going to be resistant to it. And for something like gonorrhea, it's very difficult to test for it before you start giving the antibiotic. So the doctor's going to go with his first choice, and always says, "Oh well, we're used to using drug X if this person has gonorrhea, and if it's already resistant, then you have to go about 2 weeks and wait for it to show that the antibiotic didn't have any effect." And again, that's going to happen in about 10% of the cases, that the antibiotic will just not be effective. Here's a table showing the years where we started getting different kinds of antibiotic resistance. And one of the things I think it shows well is that, so something like Staphylococcus aureus, antibiotics didn't even come into play till about 1945. So, at the start of this timeline, there weren't even any antibiotics around, but yet we found resistant strains very quickly after we started giving this antibiotic. And just a whole number of these, okay, such that by about 1990, we have so many bacteria that are resistant. Antibiotics, in a way, are becoming useless because there's just so many different types of bacteria that are resistant to them.